Inside your head right now, a highly specialized barricade is working overtime. This is the blood-brain barrier, a dynamic, living fortress designed to let nutrients in while keeping toxic molecules out of your most critical organ. It operates in a tight three-part harmony. You have endothelial cells forming the primary wall, parasites wrapping around them for structural support, and astrocytes anchoring the whole system to the neural network. Together, they maintain absolute pristine conditions. But this system has a severe vulnerability. It requires constant oxygen. If a stroke or major trauma cuts off that oxygen supply, a state called hypoxia, the cellular barrier panics. The tight junctions holding the cells together physically break apart, allowing chaotic, toxic molecules from the bloodstream to flood the delicate brain tissue. Once that physical collapse happens, doctors are largely out of options. Modern medicine currently has no targeted therapies that can directly rebuild a broken blood-brain barrier. Patients are left permanently vulnerable to severe neurodegeneration. Researchers are now looking at a different kind of intervention, called photobiomodulation, or PBM. It relies on specific wavelengths of red and near-infrared light to treat cellular damage. Pointing a red light beam at a person's head to physically rebuild a microscopic cellular wall sounds exactly like an infomercial pitch or science fiction. But this light therapy is already FDA approved for treating things like sports injuries and skin conditions. And recent lab data shows it actually aids neurological recovery after an injury. The question is exactly how. If we can prove the precise molecular mechanisms this light uses to repair a broken cellular wall, we could provide a roadmap for targeted vascular repair. To test this, researchers built a miniaturized human blood-brain barrier in a dish. This diagram outlines their trans-well model, starting left with cell cultures. By layering endothelial cells, pericytes, and astrocytes, scientists created a triculture environment to measure barrier integrity in real time. Next, they introduce the insult. They suffocate the lab-grown cells, dropping the oxygen level to just 1. To show this is a specific mechanical fix, researchers had to bait inside the cells and isolate the exact molecular trigger responding to the light. The biological cascade starts here. Red light penetrates the cell membrane, absorbed directly by the mitochondria cytochrome C oxidase. This instantly boosts cellular energy, ATP, while dissolving toxic reactive oxygen species accumulated during suffocation. The critical discovery happened in the endothelial cells. This technical graphic illustrates their response to oxygen starvation. They release a clotting protein, VWF, which drives barrier breakdown. But as the downward red arrow indicates, light therapy suppresses its production, acting as a biological switch. To confirm this specific pathway, the scientists ran a final test. They manually knocked out the VWF gene in the cells using interfering RNA, completely removing the protein to test the result. Without the VWF protein present, the barrier healed its electrical resistance exactly as if it had been hit with the near-infrared light. The light serves a specific biological function. It deactivates the cell's internal alarms, allowing the barrier to close and return to a state of homeostasis. This study moves PBM from an observed phenomenon to a mapped biological sequence. The results suggest it operates as a precise biological modulator, moving it out of the realm of alternative therapies and into rigorous science. By actively dialing down specific thromboinflammatory signals, like VWF and another prothrombotic protein called PAI1, the light specifically rescues the crucial endothelial cells that line the brain's blood vessels. In this lab-grown model, these findings suggest that barrier breakdown is not an inevitable outcome of hypoxia, but rather a reversible cellular state that we may one day be able to control. For patients suffering from acute strokes, traumatic brain injuries, or chronic vascular dementia, this insight offers a tangible path forward. The current medical status quo relies heavily on systemic drugs. Those chemicals often carry severe side effects, and ironically, they routinely fail to even cross the blood-brain barrier they are meant to treat. We are looking toward a near future where doctors can physically protect and repair the brain's ultimate biological fortress from the outside in, and they will do it simply through the targeted application of specific light frequencies. This research bridges the gap between physics and biology. It takes a simple beam of light and turns it into a highly specific, foundational tool for the future of neurovascular medicine.